Okay, so in the syllabus, right, I need one of those t-shirts that says, read the syllabus, it's in the syllabus, right, one of those teacher shirts. I gave you guys a calendar for the entire semester. It has all the classes, right, so make sure that, of course, you're looking at Tuesday, because that's when your class is, right, for dates. And then I don't know if you guys noticed, I've been adding the dates to Canvas as well. So this week is gram stain and capsule stain and how to make and test a hypotheses. There were some videos, hopefully you watched them. There's also some animations. One of them I'm going to go over in lecture today. So I've already got that open in another tab. I did make one small goof on the handout for today. If you guys flip to the very last page, you'll notice it says Lab 4. And I don't want you guys to get confused, so just cross that out. And today is obviously Lab 5. Someone else had added those pages to our documents and didn't link the header pages. So when I changed it on one, it didn't change it on all of those. I've figured out since then how to fix that. So you'll notice it's fixed on the one you picked up today. So for next time, again, there's some videos. Next time, I will not demo anything, right? I won't demonstrate. I won't do any of the stuff. At this point, right, you guys should be able to follow instructions and do it on your own. If you need to see somebody doing it or doing procedures very much like it, then watch those videos that I have posted for you. The other thing for next week is, again, of course, the following week is the exam. So we're going to do some review. Part of that review is we're going to go over this document known as staining ID. Of course, color matters, right, when you're identifying stains. So I don't print this one out for you guys because I only have access to black and white printing, which is fine for you too. If you print it out in black and white, you can look at it online in color, right? But if you want to have that document, that PowerPoint ahead of time to make notes on, if you like that, I did make it available to you, and actually I put it in a PDF so it's in the printable document format, so you shouldn't have any issues printing. Any questions on that? Okay, so if you want that for yourself, you need to print it yourself. Next week's quiz will actually be a practical quiz, which means you will do the gram stain again for your quiz and focus your slide under the microscope. The only step you won't do is make the slide. I will give you a pre-made heat-fixed slide for gram stain. You'll write out the gram stain procedure, you'll actually stain your slide, and then focus it under the microscope for next week's quiz. Any questions regarding that? So uber important that you're on time for the quiz, because if you're not on time, there's no makeup. Okay? All right. So let's start with today's lecture. So we're going to talk about if-then statements, basically forming hypotheses and testing them. I'm going to go over the gram stain and the capsule stain. So how do you make and test a hypothesis? For the most part, you set up if-then statements, right? If this is true, then this must also be true, right? Kind of cause and effect. You do this all the time in your everyday life, right? More than you even realize. Typically, we want one that can be tested. And we could write this out mathematically as A than B, right? So if A is true, then B must also be true. So one morphology equals one species on an agar plate. So if you see one color, one shiny, round colony on a plate, does that mean that all those colonies that look like that are all the same species? No, right? But we could test that hypothesis, right, by testing those ones that look alike. And as you guys will find out and see as we move throughout the semester, if it wasn't for the label on the tube, you wouldn't know one organism from another looking at the growth on the slant or on a plate or in a broth. So we could write this also upside down and backwards, which basically says if you have greater than 2 species, you have greater than 2 morphologies. Again, is that true? If you have more than 1 species, are you going to have more than 1 morphology? Not necessarily, because we just said some of them look the same, right? But they're not the same, right? So both these statements are false. So a hypothesis is true when A equals B is true, or A equals B is false. You can't have one be false and one be true, right? Either both the statements have to be false or both of them have to be true. They have to match up. And they don't for this, right? We already know, and you'll experience as we go throughout the semester, we would disprove this hypothesis that 1 morphology equals 1 species. Or 2 always are going to equal 2 different morphologies. We have a mixed culture of at least 2 species, but we only see 1 morphology, right? We can't accept either of these statements. Make sense to you guys? You do this all the time, like I said, in your everyday life, and lots of different professions too do this. Lots of different ways of writing it. So later on, almost the last experiment that we'll do, a little bit more complicated hypotheses that fluorescence will occur only if expression of the green fluorescent protein gene occurs, right? So we're only going to see color if the DNA that we give the bacteria is actually expressed, right? Turned into messenger RNA and then into a protein. Can we test that hypothesis? Yeah, and we will. And just about from now on, and in future semesters, I'm toying with the idea of, at this point, giving you guys your unknown organism and actually start staining it right from the beginning. But if you don't know what it is, you've got to test to figure out what it is, right? So that's what you guys are going to start doing. You're going to take organisms and you're going to run tests on them, right? The very first one we're going to do today is the gram stain, as well as the capsule stain. You can even look at what we did last week as hypotheses testing as well too, right? Trying to figure out what shape and what arrangement your organism was. Most of them I told you ahead of time, right? We're going to get to the point where you're not going to know. So scientific method, our very daily lives we use it, right? We form a hypothesis. If this is that, then this is that, right? If you go out in the morning and your car doesn't start, do you just go, oh well? No, immediately you start asking questions, right? Is it the battery? Right? Is it the alternator? It's a more high tech. Is it the starter, right? You know. Yeah. You go through, you start automatically, right? What could possibly be wrong with it that would contribute to it not starting? Right. It's a progression, right? And you test each possibility as you go, right? And so the same thing with your unknown, right? What shape is it? What gram reaction is it? Right? And all this data we can gather to figure out what we're dealing with. Engineers, mechanics, plumbers, right? And again, like I said, our everyday life, we use scientific method all the time, right? What do we think is wrong or right? How can we make this happen? How can I test it? How can I prove it? So Most journal articles, scientific articles, are set up in the way of the scientific method, which makes sense, right? They have an introduction. They're starting out with forming their problem and setting up their hypotheses, right? Based on prior knowledge, right? We all have prior knowledge as far as the car goes, right? We know that if it doesn't start, that potentially could be the battery. We could go and have that tested. So why am I doing these experiments, right? Method. How are you going to go about it? And this is all the procedures and protocols that you guys have been writing out, right? For most of this, you're not having to come up with this. You're using what's already been done before you. And that's fine too. The important thing is you have to know why you're doing it, how to do it, how to perform it correctly, and whether it will satisfy your needs of proving or disproving your hypotheses. Right. Results. Well, once you've done it, what are the results? What is your data, basically, right? Which you guys have gotten some practice in recording that already, writing that down. That's probably just as important, if not the most important step, right? Actually recording your data. So we record it in multiple different ways, right? Not your data, right? Because what's the whole point of doing it? Because you don't know, right? When I give you your unknown organism, are you going to know if it's gram-positive or gram-negative? No, you're going to have to do the protocol. You write out the protocol ahead of time, right? But then you're going to stain it, and then you're going to get a result, right? How are you going to record that result? Draw a picture, yeah. Maybe even write a description. Maybe even take a picture, right? Depending on what type of data, too, you might have graphs, right? Or tables to organize all the information that you gather. And then we've got to analyze it, right? And of course, if you do the gram stain and your controls that are supposed to come out positive and negative don't come out correctly, are you going to trust what you got for your unknown? No, you're not going to record that data because it's inaccurate, right? You'd have to throw that out and do it again. So we only throw out stuff when we know we've made a mistake. Right? We don't just leave it out because, it doesn't look good, it doesn't fit. But obviously, there have been times where they were trying to do one thing and discovered a totally different thing, right? In that case, you go in a different direction and you report it and you work on that. So, what should I record and how should I report them, right, is what we're thinking about as far as results go. And then what do the results mean, right? Justify any ambiguous or contradictory results, right? Why do we think we got what we got? Draw conclusions from that. So what does this mean? So definitely ask yourself this question, right? When we get the result of gram-positive or gram-negative, what does that mean? Why is that information important? So good old Graham, and that's why it's called the Graham stain, because his last name was Graham, right? And so he was playing around in the lab, doing different stains, and found out if he followed this particular protocol that he came up with, he got 2 different colors, right, when he stained different organisms. So it's a 4-step procedure. In between each step, we're going to rinse with our deionized water. I am going to demo this, right, because this can be a tricky one, but this is probably the most important staining procedure we do in microbiology, right, because it gives us some really good information. The first step is going to be using crystal violet. This is a basic dye, so what charge does it have? Positive, right? And so what's it going to stain, the cell or the background? We're going to stain the cell, right? We're going to do this for 1 minute. Remember the good golden 1 minute. If you're in doubt, do it for at least a minute, right, in microbiology. So you'll flood your whole slide like we did last week, and you're going to rinse it off, but we're not going to be done yet. The next thing you're going to apply is iodine Is this version of the lab manual still say for this step grams reagent? What did you guys write for? It does say grams iodine? Okay, good. All right. In the past, it just said grams reagent. Students didn't know it was iodine. But yeah, so we're going to add iodine. This is a mordaulant. This adheres to the crystal violet, and we'll talk about that as we go through the specific molecular steps of this procedure. So again, 1 minute, rinse with water, right? Tip your slide, rinse. The next step is decolorization, which we use 95% ethanol for this step. And this is the tough step. This is the differential step. This is where we're going to be removing color from one group of organisms and not the other. This needs to be done drop-wise until it just starts to go from dark purple, the drops, to light purple. And so this is, like I said, the crucial step that I'm going to demo and show you guys. And then you'll notice that there were 2 videos for gram staining. One of them where the audiovisual department here at Delgado came in and actually filmed me, and I think they did an excellent job of capturing that step where you could actually really see it. One of the other instructors liked my low-tech version too, and so that had good points that I should still post it, so I still posted that one for you, where he and I did it with my camera. The last step is adding the safranin, because remember we remove the color from one group of organisms, so we've got to color them back in order for us to be able to see them. So safranin's another basic dye, so what charge? Positive. What are we staining? The cells or the background? The cells. So again, 1 minute, rinse. Remember, even after decolorization with ethanol, we definitely want to rinse with water. After this step, rinse with water. Then we're going to blot dry. And hopefully for your guys' quiz next week, it'll look like this. Right? We're going to have rods that are what are called gram negative, which are pink. Because safranin's a red dye, but they get damaged in the process, so they don't stain very bright red. They come out a lighter shade of red that we know is pink. And then you should have cocci, which are gram positive. The same is true today, except you're going to separate the 2 organisms in 2 different circles on the same slide. Next week, I'm going to give you a mixture where they're going to be mixed together like this, so that you just have to focus on the one circle for me to be able to look at it for grading your quiz. So same is true for today. The rod that you're working with is gram negative, is going to come out pink. And the cocci are going to, both the cocci that we work with, staph and micrococcus, are gram positive. So they're going to be purple. Little cocci. So bacteria can be either gram positive or gram negative, and it's the color that we're referring to. Whether they stay purple means gram positive. They stay purple through the whole procedure. Or they lose their color and they get counterstained with the safranin and come out pink, they're said to be gram negative. Their shape doesn't have anything to do with what color they come out. Okay? So notice we have bacilli that are gram negative, like we're going to be working with today. There are some cocci that are gram negative. Unfortunately, a lot of these are very pathogenic. And so we actually don't work with any of them in this lab. So you actually won't see any gram-negative cocci. I'm going to try and find some non-pathogenic ones for us to work with. But things like Neisseria gonorrhea that causes gonorrhea, and Neisseria meningitis that can cause a pretty serious form of meningitis, are gram-negative cocci, and they actually tend to form pairs. So you could say they're what? What would be pairs of cocci would be the term for that arrangement. Diplococcus, yeah. So that's something they look for, right, when they're looking for those particular types, right? That's a pretty good clue in when they do a gram stain and they see gram-negative cocci in pairs. They're like, ooh, it might be Neisseria, so let's do some further testing. So E. coli, which is the one we're going to use today, is gram-negative. Klebsiella pneumoniae, which is a pathogen, which we don't use anymore, are also gram-negative rods. All the cocci we work with in this lab currently are all gram-positive. So they're all going to come out kind of purple. And of course, the staph are going to cluster in the micrococcus. You guys remember the characteristic arrangement for micrococcus? It's 4 of them together. What do we call that? tetrads. And then we have some gram-positive bacilli. And in fact, all the bacillus that we work with in this lab are gram-positive. So they're rod-shaped, but they'll come out purple when you stain them. So when we gram stain, it's going to either come out purple or pink. And in general, this divides the known bacteria in about half. 60% are about gram-negative, and the other about 40%. Did I do my math right? Yeah, 40 are gram-positive. It's early still. But what do we really know when it comes out purple or pink? What we know is its cell wall composition. Because it's the cell wall that determines whether it's going to stay purple or it's going to get decolorized and be pink and be what we call negative. So gram-positive bacteria, if you were to look at them using like an electron microscope, you would see a really thick layer of cell wall that's made of a substance for bacteria known as peptidoglycan. And then also associated with that are other molecules represented here by little yellow squiggly lines known as tactoic acid. It's almost like rebar is for cement, right? It helps hold this really thick layer of peptidoglycan that makes up the cell wall. In contrast, gram-negatives will tend to have a very thin layer of peptidoglycan, and then they have an additional membrane known as the outer membrane. So in addition to the cell membrane or plasma membrane, they have an outer membrane. And this outer membrane is unique in that the very outer leaflet isn't made of phospholipids like we're used to. It's made out of lipopolysaccharides, commonly abbreviated LPS. And so that's why you see little squiggly lines coming up, because they actually have sugar associated with them. The cell wall is really important for bacteria. Most of them have them. It's very rare for them not to have it. It helps them resist osmotic pressure. When water comes rushing into our cells, if we're in a hypertonic solution, what happens to our cells? Hypo, right? They'll swell up and burst. Yep. And so these are unicellular organisms. Can they afford to burst? No, because they'd be dead, right? Because they're one cell. That one cell dies, they're dead. For us, we've got lots of cells, right? So, you know, if we kill a few in a hypotonic solution, it won't be the end of the world. We've got cells to spare, right? So there's that tactoic acid, as I said, the yellow squiggly lines, almost like rebar. So for peptidoglycan, and again, you'd need an electron microscope to see this, because remember the range of our microscopes is only about 200 nanometers. Although this is pretty thick, it can range from 30 to 100 nanometers thick for gram-positive, this peptidoglycan layer. Oops. So yeah, that just says, right, not within the range of our microscopes, right? If you're lucky, you'll see a thin little line. So peptidoglycan, also known as murine, is a polymer, right? So it's repeating subunits of two sugars, two modified sugars, commonly abbreviated NAG and NAM. So you have your N-acetyl glucosamine, and N-acetyl muric. And these are alternating sugars, right? So imagine a jawbreaker, right? Layer after layer of sugar. So that's what these guys are coated in, is these alternating sugars. Layer after layer after layer. Now each one of those layers is stuck together by amino acids. That's the peptide part of the name, because the bond between amino acids is called a peptide bond. So that's represented here by these amino acids here and here. So this layer we hook to the next layer, and so on and so forth. So it's almost like mortar even between bricks, right? Holding those layers together. Stuff can easily get through this layer though, because this is more like a wicker basket, right, that you could stick your Easter eggs in, but water would go right through that, right? But the eggs are so large, right, that they'll stay in that basket. So this is going to give them the rigidity. This is going to help form those shapes that we see, but it's also help protect them from bursting under osmotic pressure. Glycoproteins, right, and glyco refers to sugar. These are sugars associated with proteins. And there are some organisms that have this type of substance associated with their cell walls as well. It's very common for them to add sugars to their proteins. So why is a crystal violet iodine complex bigger than crystal violet alone? So if we look at the structure of crystal violet, it has chlorine ions associated with it when it's in its solid form. When we put it into solution, the chloride ions separate from it. When crystal violet goes into the cells, because notice it's positively charged, we then add iodine. And the iodine will complex, because again, another negatively charged ion, with the crystal violet positively charged dye that's on the inside. It's now bigger than it would be with the chlorine ions, because if you look on the periodic table, iodine is bigger than chlorine. Just simple chemistry. It's bigger, right? So we end up with a bigger molecule now on the inside of the cell. So we won't get into the specific chemistry. So I love this kitty. This is one of the instructors who teaches this class. His cat that was a stray that adopted him. You know how that works. So this is Sachimo. And he squeezes between the fence and the gate there. And after he eats, he can't leave. Why not? He's too fat. Just that little bit of food, right, in his stomach, he can't squeeze through anymore, right? So I want you guys to really think about and visualize that when you think about the crystal violet and the iodine, how it's a much larger molecule, right, and how it's not going to fit back through the original hole that it went through because it's bigger, just like Sachimo. Make sense? So those of you guys in lecture have seen this before, right? So this is a little toy. I wanted just that one to show up. There we go. I guess not. Get this one animated differently. It has these different shaped holes, and inside are the blocks, and those blocks barely fit into those different size shaped holes. So little kids put the blocks in, and they don't know that at the top and the bottom are handles, and you can pull apart these two sections and dump out all the little blocks so you can do it again. So instead, they stick their little hand in the hole and try to get the blocks out. Doesn't work, right? Because that block barely fit in there all by itself. Now, you've got a little hand in there with the block trying to get out. It's not going to happen, right? It's too big. The hand and the block combined isn't going to happen. So you have to convince them, to let go of it, stop screaming, right? And then you teach them how to open it, and it's all good. For the procedure we're doing today, this is very similar, right? Crystal violet goes in, and then iodine goes in, and they combine. Same thing here. Block went in by itself, then little hand went in, but then they combine on the inside, and they can't get out. There's one other thing that happens with the gram stain, though, is not only do we make a bigger molecule, crystal violet and iodine, this hole shrinks in the procedure that we do. So you imagine if that happened to the kid, they stick their little hand in there and then the hole shrinks up around them, you really have a screaming kid. Alright, so here's your guys' intelligent quiz for today, but those of you guys in lecture can't answer it because you already know the answer. How do you get this ball out this hole? Think it went in like that? No. How are we going to get that ball out? Take the top off? We can't take the top off. Take the sticks out, right? Take the little pegs out, right? Then the ball will fit. So clearly the ball went in and then they stuck the pegs in, right? That trapped it in. So again, I want you guys to visualize that crystal violet and the iodine getting trapped inside our cells. So there's an animation. There's a video animation that's really good, but then there's another animation that I want to go through. And so it's under the videos link for Lab 5. So I just opened that on a separate page. And it's this link right here. It kind of gets lost. So thank you to these lovely gentlemen that put together this awesome animation. There's no audio to this though, right? So I'm kind of adding my own audio, but it is scripted. So we're going to choose gram-positive first. And so here's the script down below. So this represents what you would look like if you were at an electron microscope, right? We're looking at the molecular level of a gram-positive cell. So you have the cell membrane, right, made out of phospholipids, right? Phospholipids with embedded proteins. Then we have the peptidoglycan cell wall layer that's pretty thick. The teichoic acid, right? Our crystal violet iodine complex, our decolorizer, right? These are all the steps of dyes that we're going to use, the crystal violet by itself and the iodine by itself. So the first step is what? Add crystal violet. For how long? 1 minute. What do we do after the minute? We rinse off our slide with deionized water, right? So remember, it's positively charged inside. The cell is DNA, it's negatively charged. The positive and the negative are attracted to each other, those opposites. The crystal violet, right? It's a positively charged dye. So it goes inside. So at this point, the cells will be purple. Then we're going to add iodine, which, if you know from biology class, also complexes with starches and turns black, so our slides may look a little black. Don't panic. It's okay. So that's going to go inside, and it's going to complex with the crystal violet, right? So we get that larger molecule. Now what's the next step? We did crystal violet, we did iodine, we rinsed with water, decolorized dropwise with ethanol, right? Watch what happens to this cell when we decolorize.
That's the ethanol that causes that?
Mm-hmm. So what changed? What is this stuff? Cell wall. Peptidoglycan for bacteria. It did what? It shrunk. So you notice the spaces between the molecules also got smaller. So is the crystal violet iodine going to be able to get out? No. It's like Satchmo. He's stuck, right? Or the little kid in his hand. It's not going anywhere. We're still going to do the last step of the procedure because you don't know, right? Until you get done and look at it on the microscope whether it's gram-positive or gram-negative. So we got to do the last step, which is stain with safranin. So that's going to go in. But if you ever did laundry, right? If you've got purple and you had red that ran, are you going to notice that the red ran on the purple clothes? No, right? The purple is the more vibrant color. So even though we stained it again with red, it's still going to look what? Purple. So gram-positives stay purple, right? And positive is just the result, right? Saying that it stayed purple. So now let's go through gram-negative. Different structure here, right? So we've got cell membrane, or plasma membrane, We've got peptidoglycan, but it's thinner, huh? And what's this stuff? What is that? What do we call this layer? Nope. It's another membrane, but it's the outer membrane. It's not that hard. Thank goodness, right? They didn't come up with some crazy name. It's just the outer membrane. But the important thing here is this layer right here within that outer membrane is a different substance than most membranes are made out of. This is lipopolysaccharide. So it's got the lipid component, the phosphate group, but then it has sugars associated with it. And this is actually what makes gram-negative cells so toxic, and why, again, doctors want to know if you have a gram-positive or gram-negative infection sometimes, right? Because this can be really bad. So again, all the steps, right? These cells are going to go through all the steps. So first step, you got to know it for the quiz next week, which is why I'm going to keep repeating it. So crystal violet goes in, and that's because these cells are dead, right? Remember, we're going to heat fix them onto the slide, right? We're going to kill them. We're going to add the iodine. It's going to complex. We're going to rinse with water. Now the decolorization, this differential step for this procedure, what differentiates gram-positives from gram-negatives. What's going to happen? Two things happened, right? The outer membrane went goodbye, right? Or severely damaged. And the peptidoglycan layer shrunk, but it wasn't very thick to begin with, right? So is the crystal violet iodine going to stay in this cell? No. Out it goes. So if you were to stop here, the cells would be colorless. But we want to see them, right? So we got to counter stain with, what's the last stain? Safranin. And so it's a red dye, but they're going to look pink because they're not going to retain a lot of the dye from the damage that they underwent under decolorization. So under the microscope, they will look pink. So again, you need to know each one of the steps. A table is a great way, right, to organize information. So gram-positive cells, first step, basic dye, positively charged, it's going to go in. All the cells are going to be what color? Purple. Then we add the iodine. It's going to complex with the crystal violet. Right? Color-wise, they're pretty much still going to be purple if you were to stop and look at them under the microscope. When we do the decolorization step, are they going to decolorize? Gram-positives. No, they're going to stay purple because the peptidoglycan layer that's really thick is going to shrink and trap in that crystal violet iodine, that larger molecule. Then we're going to stain with safranin, but remember they're still going to look what color? They're still going to look purple. What about for gram-negatives? First step, add crystal violet, they're going to be purple. Next step, iodine, they would still be purple. This is the different step, right? Are these guys going to decolorize? Yes, right? Because they have that thin layer peptidoglycan, can't trap it in, and that outer membrane is going to be disrupted. It's going to allow the release of the crystal violet iodine. So they would be colorless at this point if we stopped. You would not see them. Yeah, the last thing we add the safranin so that we can actually see them. And again, they're going to look pink, not red. So the two of them side by side, the really important step, right? Decolorization step. That's where they're going to differ, right? And where we're going to get the different results. So just drawn out, right? If we looked at two side by side, just look at the colors, right? So they're all going to be purple. First step. Iodine, they're all still going to be purple. Then when we decolorize, we're going to make the gram-negatives colorless. When we stain with the safranin, we're actually going to end up with a darker purple. And then, of course, the light pink for the gram-negatives. So now it says let's predict some mistakes you could make. So we're going to consider each mistake separately. So you do all the steps right except for this one step you mess up. So for review, 4 major steps. First, dye, crystal violet. What's the next one? Iodine. Decolorize with ethanol, dropwise. Last step, stain with safranin. Okay, so what if you don't add the iodine for a gram-positive cell? So let's look at each one of the steps. First step, crystal violet. They're going to be purple, right? What if you don't add the iodine? What do you say, Mike?
This is all.
Yeah. Okay. What happens if you don't add the iodine? What doesn't form? The crystal violet iodine complex doesn't form. We don't get a big molecule, right? We just have iodine all by its lonesome. So when we decolorize, are they going to stay purple? Nope. Molecules are tiny. They slip out. So now they're colorless. So then we counterstain with safranin, and they come out pink. Oops. What did I tell you it was? This is your control I gave you that is gram-positive and supposed to come out purple, and yours looks pink. Is that what you're supposed to get? No. All right. If it's a gram-negative, will it matter you screwed up this step? I don't know. Let's see. Crystal violet. No complex, right? Decolorize. Well, it's supposed to get decolorized, right? So it'll still get decolorized. And then we're going to stain it with safranin, and it's going to come out pink. So you're good, right? Not a problem with gram-negative. The problem is what color is your gram-positive? Pink. Right? Both your gram-positive and your gram-negative on your control slide are both pink. Can you trust your result for your unknown? No way. What if you don't decolorize? So for gram-positives, we're going to do crystal violet purple, iodine. We're not going to decolorize. So they're going to stay purple. We're going to stain with safranin. There's going to be a dark purple. Is this the normal result for gram-positive? What color are they supposed to be? Purple. So no problem here, right? The problem is if you do that with gram-negatives, they're purple, and you don't what? You don't decolorize with the ethanol, they're still going to be, you don't decolorize, they're still going to be purple. And even if you counterstain with red, they're still purple. Are they supposed to be purple? No. So again, if your control slide, both circles are purple, you messed up, right? Because one's supposed to be pink and one's supposed to be purple. So maybe you forgot to decolorize. What if you don't add the safranin? Gram-positive cells. Is this going to matter? Well, you start out with crystal violet, they're purple, right? You add the crystal violet, they're still purple. You decolorize, they're still purple. You don't stain them with safranin, but they're still already purple. Are they supposed to be purple? Yes. They're just going to look a lighter shade of purple than normal. Gram-negatives. Crystal violet, iodine, you decolorize, and you basically stop here. Are you going to even see these cells? No, at this point they're what? They're colorless. So if you don't do the safranin, right, so you end up with one purple circle and one, I can't see anything. What did you probably do? You forgot the last step, right? You didn't do the safranin because you can't even see them. So we're not going to make any of those mistakes though, right? No. All right, this one's interesting. I actually did this just to see for ha-has what would happen. So what if you reverse the order? What if you started out with safranin instead of crystal violet? So there's a reason why we start out with the purple one, right? So for gram-positives, what would happen? We would first stain them red, right? Then, believe it or not, the iodine, I think, complexes. We wouldn't decolorize them. Then we'd stain them with the purple. So now they're really dark purple, right? Is this normal result for gram-positives? Yeah. The problem lies with our gram-negatives. So if they start out red, and we remove the red and they become colorless, and then we stain them what? Purple. Uh-oh. Are gram-negatives supposed to be purple? No. And what did we say our gram-positives were? Purple. Do you know if you put them side by side, you can tell them apart? Because what do we know about the gram-positives? They got stained how many times? Twice, right? Crystal violet and safranin. They actually end up being a darker purple, and the gram-negatives a lighter purple. Would you want to try and figure out whether you're seeing dark purple or light purple? Yeah, no. Y'all, purple and pink is hard enough. You'll soon find out. Alright, so summary of mistakes, right? No iodine, everything's going to be pink, right? No decolorizer, everything's going to be purple. If you don't do your safranin, you're not even going to see your gram-negative cells, right? And if you were to reverse the stains, you're going to end up with dark purple versus light purple, which would be a nightmare. So, we use these terms way back first lab, right? So, false negative. So, gram-positives are supposed to be what color? Purple. So, if they come out pink, which is negative result, and this is your control slide that you knew was supposed to come out positive, this is a what? False negative result. Right? Because you're seeing negative, but it's supposed to be positive. So what would we say is the problem here where gram-negatives are coming out purple? That's a what result? What's purple? Positive, right? So this is a false positive result. And same is true for reversing the stains. Worst case scenario is if you don't do the safranin, right? And you end up with undetectable cells. You won't actually even see them. So both crystal violet and safranin, and we used crystal violet last week, and that was my little private haha joke because we stained E. coli purple. And E. coli, when you gram stain, it's going to come out pink. It's gram negative. So that's why I said it's the only time it gets to be purple. All the rest of the time it comes out pink. So we're usually gram staining it. So how is it that those are both basic dyes, but we end up with different results? Because we're not just simple staining, are we? We're doing a lot. We're doing crystal violet, we're doing iodine, we're decolorizing, and we're using safranin. And that's the key there. That's why this is a differential stain. That's why we end up with two different colors, is because we have a decolorization step, right? That's what we were trying to get at here. So anytime you have two different results, you're usually doing a differential stain, and at some point you're doing a decolorization. With the endospore staining that we're doing next week, we use water, something as simple as water, to decolorize the cells. and not the endospores. So it's still a differential technique. You're either going to see spores or not. So if we were to look at eukaryotic cells, right, our own cells, we would take a scraping from the gum line. Would we be gram-positive or gram-negative? Well, let's think about this. Gram-positive. So first thing is crystal violet. Next thing is iodine. We get that complex. The next thing is decolorization. Why do gram-positives stay purple? What happens to those bacteria? The crystal violet iodine complex forms, but when we decolorize, what happens? We shrink what layer? The peptidoglycan, the cell wall layer. Who has peptidoglycan as a cell wall? Just bacteria. Do we even have cell walls? No. Our cells don't have cell walls, right? So, one, it's not peptidoglycan, it's not going to shrink, and two, it's not even there. There's no cell wall. So if there's no cell walls, the crystal violet iodine going to stay inside? Nope, gets washed out. Then we counterstain with safranin, and they're going to be pink. So this big fried egg looking thing that you've seen before, that's one of our squamous epithelial cells that would be found in our mouth. And there's the nucleus. That's another clue that it's a eukaryote, right? And then all these little purple things are gram-positive bacteria that are in our mouth. But yeah, we'd always come out gram-negative. You can stain anything using the gram stain, and you're going to either get purple or pink based on how those dyes react with those cells. We primarily just use it for bacteria because we're really concerned, as I said, about those gram-negatives because they're toxic. Some gram-positives are pretty bad guys too, right? But it's going to let us know what we're dealing with. So I'm not going to go through this, but remember, for each one of these, you could go through the scientific method, right? We can start with the hypotheses. We have a method, right, a protocol that we follow. We're going to get results. It's going to be purple or it's going to be pink. We can trust those results if they come out as expected for our controls. And that's basically what you're doing today is controls. You're going to know what they're supposed to be. If they don't come out right, we got to figure out why, right, and what that data means. And for us, remember that that really tells us about the cell wall, right? If you're gram-positive, you have that thick peptidoglycan layer. If it's gram-negative, it's the thin layer and an outer membrane. And as I said, it's about 60-40. So the next thing that we're going to look at today, and I've divided up the work this semester with this so that each one of you, but if there's only like 2 or 3 of you guys at the lab bench, then somebody needs to make an extra slide. But there's 4 different organisms that we're going to stain using the capsule stain procedure. And a capsule is an addition to the cell membrane and cell wall. This is an additional layer. So if you have the cell, right, and then you have the membrane and you have the cell wall, outside of that even another layer would be the capsule layer. The problem with this is the location of this layer and the fact that none of the stains that we use stain it because it's not a charged molecule usually. It doesn't have any charge to it. So remember, our dyes are charged, either positive or negative. It's not going to attract either one, right, because it's not charged, positively or negatively. So they're sometimes referred to as a glycocalcics, referring to glyco meaning sweet or sugar, calcics referring to husk or outer covering, right? So these are like sugar-coated bacteria. So most of them are polysaccharides, and they're outside, as I said, the cell wall. Occasionally they're made of glycoproteins, like I mentioned earlier. And there's only one that's exclusively made out of amino acids instead of sugars, and that's actually anthrax. Anthrax's capsule is made out of amino acids instead of sugars. So it could be a capsule, which is really nice and uniform, or a slime layer, which is more diffuse. But again, it's the outermost layer if they have this layer. Where the capsule tends to be really firmly attached and well organized. And that's what we're looking for today. You see this feature a lot in what we call pathogenic bacteria. Although again, we're not working with any pathogens, others do have it. Because it allows them to stick to our cells, so that it allows them to adhere, as well as it allows them to basically be disguised from our immune system. They are able to evade our white blood cells that phagocytize, that engulf and eat bacteria. So really important, there's some steps in your protocol that you probably wrote down that we're not going to follow. So we are going to put the Congo red on the slide and mix the bacteria in. So this is very reminiscent of what we did last week. What staining procedure started out with the dye on the slide and adding the bacteria. The negative stain, right? So we're basically going to start out like a negative stain. We're going to use the edge of the slide and we're going to spread it out. We're going to air dry on our light boxes. We don't heat fix this slide. Right? Then we're going to flood the slide with Menville's reagent once it's dried on our staining trays. This actually causes a pH change. So you're going to go from a red slide to kind of a grayish looking slide. There's acetic acid in this. Anyone know the common name for acetic acid you might use at home? Vinegar. So again, it's going to smell, right? Those of us that don't like vinegar, it doesn't smell good. Other people, it makes them hungry. Me, not so much. I'm trying to develop an appetite for vinegar, though. I'm getting there. But yeah, I don't have that southern quality of putting vinegar on everything. But yeah, so that's acidic. It's going to lower the pH as well. It's going to stain the whole rest of the slide. In your procedure, also you'll notice in the book it says to add a drop of serum. We don't do that step. And because we don't do that step, we also skip this step. We do not rinse with water for these slides. Just tip them and blot them dry in your book. Just tip off the excess liquid onto your staining tray and blot it dry in your book. And then remember, always oil immersion because we're always working with bacteria, right? We're going to go all the way to oil immersion. There are a couple of people that are on the naughty list, right, for having left oil on their microscopes last week. Don't continue to be on the naughty list because then you have to share that microscope with everyone else who's on the naughty list. And that's the microscopes that aren't so nice. Someone is concerned that they're on the naughty list. Oh, we had that switch up. Okay. But it wasn't either one of those numbers, was it? On the sticky note? I don't want to embarrass anybody in my recording. Okay. All right. Yeah. So, like I said, we're kind of going to negative stain, and then we're going to kind of simple stain. So I've kind of, for today, almost done like a cooking show. I've already done the first step so that I can go right to the second step. So I'll do the first step and then put it off to the side, like a cooking show, and then I'm going to take the finished product out of the oven or the semi-finished product and add the cheese or something, right? So just know that I'm doing that today, right? It's a cooking show today to save time. So what's the purpose of the serum? Why do they say they add the serum? Which we're not going to do. Helps stick the bacteria to the slide. Because believe it or not, the capsule helps them stick to our cells, but not to glass slides. So sometimes they will just slide right off, which is again why we're not going to rinse them. If you forget and you rinse, it's not the end of the world, y'all. They're microscopic. As long as you have like a tiny little speck of something on your slide, you'll see something under the microscope. But try to remember to not rinse them. Right? So they don't go sliding off. So some oral bacteria produce an extracellular capsule. What would be the benefit of that? What do we say the function of the capsule is for them? So what is it going to allow them to do? If they're in our mouth, right, in order to stay put, they need to adhere. They need to stick, right, attach. And then if they were to even get into our body, they're defended because they're like wearing a disguise, right, having this layer that can evade phagocytosis, right, and antibodies sometimes don't recognize them. So capsule stain, depending on what reagents you're using, what procedure you're using, they'll look a little bit different. Ours is going to look pretty much like this side over here, where the cells are going to be kind of purple, and the background's going to be kind of grayish. And if there's a capsule present, because one of the ones you're going to stain today, there's no capsule, you're going to see this clear halo, right, this unstained layer. Yeah, the bonus. She hasn't taken the quiz. But it's on the homework anyways, but yeah. Okay, so why is an acidic dye, why is the acidic dye Congo red used? What are we doing when we apply the Congo red? What are we staining? We're staining the background, right? We're negative staining, basically. Remember, let this completely dry before you move on to the next step. Stop anybody who tries to look at a red slide under the microscope today, because the minute they put oil on it, it's ruined, and we cannot fix it. All right, so be a good lab partner, and you're relying on your lab partners, right, because we're going to all make the 4 slides. Okay, Menville's reagent is a basic stain as well. Right, so what are we staining here? So first we stain the background, and now we're going to stain the cell, right? Everyone with me? And a pH change is going to happen so that we get that color change too, to the whole slide. Is the capsule stained by the dyes? No. So instead, we stain the cell and the background. So do you see how the background's gray and the cell is purple? Do you see a capsule? No. One of your slides is going to look like this today. One of the cocci does not have a capsule. Right? So you're not going to see the little halo. And then the other cocci is so small, it's going to be really hard to see the halo. So if we get a good slide, we'll run around and look at everybody's slide, right? We'll look at that one good slide. But that's also why one of the organisms I'm giving you guys today has a nice huge cell and a huge capsule. So we should be able to see it really clearly. So if you just did a simple stain, would you know a cell has a capsule? No, because when you simple stain with a basic dye, you just stain what? The cell. Are you going to know that there's this outer layer outside of that? No. I mean, I have it drawn in here, but you're just going to, you've already done this, right? You just saw cells. You didn't see anything outside of the cell. What if you just stain the background with an acidic dye? The cell is going to be what? Clear. So do you know that it has a capsule? No. I mean, if you compared a simple stain to a negative stain, you might notice it's bigger. But remember, what happens when we simple stain that changes the size of the cell anyways? Yeah, when we heat it up, we shrink it. When we heat fix it, right, to stick it to the slide, we shrink it. So that wouldn't be reliable, right? You've got to do both steps, right? Because you're not going to know. So there's our scientific method. And the glossary of terms is at the end. And yes, this PowerPoint is posted. Right now. and I will turn on the drawings to the answers for the ones that you'll look at today. You have one additional organism, so you're going to do 2 cocci. One's got a capsule, one doesn't. You're going to do a bacillus that has a capsule, and then you're actually going to do a eukaryote, a yeast. So these guys are going to be really big. You're going to do 2 different cocci. One has a capsule, one does not. You're going to do a rod that has a nice big capsule. And then you're going to do a yeast, which is a eukaryote. This is not a pure culture. This is baker's yeast. Okay? Good old baker's yeast you buy at the that I bought at the grocery store. Okay? And grew up in the lab. Okay? This is going to be contaminated with bacteria, and some of them even have a capsule. But you want to try and scan around and see if you can find a yeast, because they're going to be really big. And you've looked at yeast before, right? Except these yeast produce a capsule, right? We're going to capsule stain, and you'll see the nice big capsule, right, that they produce. All right, so go ahead and get out supplies.